Imagine you were alive back in the 1980s and were told that computers would soon take over everything, from shopping to dating and the stock market. That billions of people would be connected via a kind of web. That you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever.
In the 20th and 21st centuries, science and technology has greatly advanced and developed worldwide and affected many aspects of human life. Unarguably, advancement in technology over the years has made our lives easier, specifically on our health and medical sector, with the advances in research, diagnosis, and treatment have significantly contributed to both the length and quality of human life. Indeed, with the continuous advancement and progress, we can make impossible things possible. We can now detect cancer cells and make technological advancement to stop and help slow down the spread of these cells. We can also now have the in vitro fertilization, wherein mothers who have serious illnesses or are incapable to bear a child will have the chance to have a baby. More importantly, genetic engineering have also taken the world by storm. A breakthrough that is so relevant and useful to us in terms of agriculture and livestock, in research and for medical progresses. These are just some of the technological and scientific advances. But today, we will be focusing our discussion on genetic engineering. I am Geraldine Ragini, together with Eva Jane Alibin, Giselle Ann Cabalbag, and Rogeline Valencia, and we will further enhance and make you more understand this topic. So these would be the topics that we will be discussing. First, we have the introduction, brief summary, the methods and techniques of genetic engineering. We will also be discussing the application or uses of genetically modified organisms in terms of agriculture, as well as livestock in health and medical area. And lastly, we have the conclusions. Can you spot the difference of these two sets of mice based on their physical appearances? The first picture seems to be the normal mice, with its blackish brown color, yet on the other picture, this mice seems to be different. Its body seems to be glowing, especially their eyes. They look very sharp and deadly. Yet how did that happen? Why is it that it looks very different to the original appearances of the mice in the first picture? Our question will be answered as we go on with our discussion. But first, let us familiarize first what is genetic engineering. Genetic engineering, also called genetic modification, is a process that uses laboratory-based technologies to alter the DNA makeup of an organism. GMOs, or genetically modified organisms, are what we call for the product of genetic engineering. This may involve changing a single base pair of the nitrogenous bases, deleting a region of DNA, or adding a new segment of DNA. So basically, this process aims to alter or change the genetic structure of a certain organism to have desirable properties, to enhance the capabilities of an organism beyond what is normal and more suitable for a particular purpose, like developing vaccines or medicines, pesticide resistant for plants, and such. Much likely, genetic engineering is a process that involves first identifying the genetic information or a gene that gives an organism a desired trait. Next, copying the information from the organism that has the trait, and then inserting that information into the DNA of another organism, and lastly, growing the new organism. Jack Williamson coined the term genetic engineering in his science fiction novel entitled Dragon's Island. Moreover, genetic engineering, as I said a while back, have been dominating the biotechnological and medical world, as it's been widely used to the production of cancer therapies, brewing yeast, genetically modified plants, livestock, and more. Let's move on to the brief history of the development of genetic engineering. So in 1972, Paul Berg created the first recombinant DNA molecules. Paul Berg is the father of genetic engineering, as he is the first person to acknowledge and make use of this biotechnology. With the aid of lambda phage, he was able to insert the SV40 virus gene into the bacterium. In 1973, the first genetically modified bacteria was created by the biochemists Herbert Boyer and Stanley Cohen. They developed genetic engineering by inserting DNA from one bacteria into another. In 1973, Beatrice Mintz and Rodolph Janisch created the genetically modified mice, the first genetically modified animal, by inserting a DNA virus into an early-stage mouse embryo and showing that the inserted genes were present in every cell. In 1982, the first commercial development of GMOs, or the insulin-producing bacteria, to treat diabetes was developed by Eli Lilly. She prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them into the plasmids of E. coli to produce insulin chains. In 1986, the first trials of genetically engineered plants occurred in France and USA. Tobacco was tested as herbicide-resistant plant. In 1994, they began to sell genetically modified food. The first GMO produced, created through a genetic engineering, is a GMO tomato. It was evaluated by federal agencies and proved it to be safe as traditional bred tomatoes. In 2003, they began to sell GMOs as pets, or the glowfish. The glowfish are one of the first genetically modified plants to become popular within the pet trade. These fish were developed by introducing different fluorescent proteins into the genome of the fish at the early stages of development. Genetic engineering is not as simple and easy as we think. It is very complex, requires a lot of effort and money, and very hard to do. So we have here the methods used in genetic engineering. That includes the plasmid method, vector method, and lastly the biolistic method. The first one is the most common, which is the plasmid method. A plasmid is a genetic structure in a cell that can replicate independently. It is also typically a small circular DNA strand in the cytoplasm of a bacterium or protozoan. The plasmid serves as a tool to clone, transfer, and manipulate genes. Researchers can insert DNA fragments or genes into a plasmid vector, creating the so-called recombinant plasmid. So, as we can see in the illustration on number 1, we have here the Agrobacterium tomophaciens bacterium, where we will obtain the plasmid. First, the plasmid is removed from the bacterium and the target DNA, which is the colored gray area, is cut by restriction enzyme. At the same time, the foreign DNA is also cut by the same enzyme. Then, the foreign DNA is inserted into the target DNA of the plasmid. So there is now the recombination on the plasmid on the target DNA. The plasmid is now reinserted back into the bacterium. Then, the bacterium with the recombinant plasmid is now used to insert the target DNA, carrying the foreign gene into the desired organism. So in this case, the plant undergoes the genetic modification. Then the plant cell, which has the foreign or recombinant gene, are now grown in a culture to multiply and grow. Then lastly, a plant is generated from a cell clone. The cells containing the foreign gene may express a new trait. The second method is the vector method. This uses a vehicle or a vector to transfer foreign genetic material from one cell to another. Vectors used are typically viruses that attach to a cell, insert its DNA into the host cell, and then detach itself. The DNA, now part of the host cell, will start replicating as part of the genetic information of the host cell and producing the desired trait. This method is said to be better than the plasmid method because it gives good results in the host when the desired features are tested. So last, we have here the biolistic method. This method is totally different from the other two methods because it is mainly related to the engineering of the plants. This is also known as a gene gun method. It uses a gene gun to fire the DNA particles into the cell. The required genes are inserted into the gun, which is made up of tungsten, gold, or silver, and the DNA particles are shot into the targeted cell. This is typically used to modify the plants.
Genetic engineers devise various techniques through many years of rigorous research and invention on gene manipulation and function. These techniques have been created to insert, modify, or delete DNA, which can be from a specific base pair in a specific gene to entire genes. Some of these techniques in genetic engineering are artificial selection, gene splicing, cloning, and gel electrophoresis. First, I'm going to discuss artificial selection. This technique is called artificial selection because people, instead of nature, select which organisms to reproduce. Breeders chose individual organisms with desirable characteristics or certain phenotypic traits for breeding. To put simply, artificial selection is the selection of preferred visible traits in animals or in plants, and then they breed these organisms to produce offspring with those desired traits. Additionally, renowned scientist Charles Darwin is credited with coining the term artificial selection in his book On the Origin of Species, which he wrote upon returning from the Galapagos Islands and experimenting with crossbreeding birds. There are three types of artificial selection. These are the selective breeding, hybridization, and inbreeding. In selective breeding, animals with desired characteristics are mated to produce offspring with desired traits. This involves the passing of important genes to the next generation. This also allows important breeders to control and fine-tune the traits. Examples of the products of selective breeding are champion race horses, cows with tender meat, and large juicy oranges on a tree. Other examples include Angus cows being bred to increase muscle mass so that we get more meat, and of course the egg-laying hen, which produces more eggs than the average hen. Also under artificial selection is hybridization. This occurs when organisms from two different species are bred together. The offspring is called a hybrid. One of the most famous hybrid is a mule, which is a cross between a donkey and a horse. Moreover, another example is the disease-resistant potato known as the Burbank potato, created by Luther Burbank, who was a botanist, horticulturist, and a pioneer in agricultural science. As a matter of fact, Luther crossed a disease-resistant potato with one that had a large producing capacity to produce a disease-resistant plant that yield a great amount of potatoes. The last type of artificial selection is inbreeding. This is the breeding of organisms that are genetically similar to maintain desired traits. Dog breeds are kept pure this way. It is how a Doberman remains a Doberman. It keeps each breed unique from others. However, there are some risks to be considered in inbreeding. Since both bred organisms have the same genes, the chance that a baby will get a recessive genetic disorder. Inbreeding also decreases variations and result in poorer reproductive efficiency, including higher mortality rates, lower growth rates, and higher frequency of hereditary abnormalities. Moving forward, cloning is also a genetic engineering technique. From the word cloning, we can already decipher that this technique involves creating an organism that is an exact copy of another. In a sexual reproduction, a group of cells or organisms that are genetically identical to its parent organism is called a clone. As a result, they will have the same exact DNA as the parent. The most famous clone was the Scottish sheep named Dolly. In addition, cloning has potential benefits. Organs for transplant into humans have been made possible. It also saves endangered species as we can now create the same replica of organisms that are on the brink of extinction. On the other hand, cloning also raises concerns. Cloning has actually low success rate, and expensive clones are imperfect and less healthy than the original animal. Cloning also leads to decreased biodiversity. There are three types of cloning. The first one is gene cloning, which creates copies of genes or segments of DNA. Gene cloning is essentially recombinant DNA technology, where a piece of foreign DNA is inserted into a vector, which can be copied by a host cell. The other one is reproductive cloning, which creates copies of whole animals. The third one is therapeutic cloning, which creates embryonic stem cells. This involves the production of patient-matched stem cells for disease treatment. So, how do we create a clone of an organism? First, we have to isolate the donor DNA fragment or gene. In here, the enzyme endonuclease is a key enzyme in molecular gene cloning. It has a specific restriction site for its action. The enzyme generates a DNA fragment either with blunt end or with a sticky end. Next is the selection of a suitable vector. When donor DNA fragment is incorporated into a host cell, it will not replicate because the isolated gene doesn't have the capacity to replicate itself. So it is important that before the introduction of donor fragment into host, a suitable vector should be selected. Then, the incorporation of donor DNA fragment into the vector is performed. The plasmid vector is cut open by the enzyme used for isolation of DNA donor fragment, which is the endonuclease. The donor DNA fragment and plasmid vector are mixed together. In the presence of DNA ligase, base pairing of donor DNA fragment and plasmid vector occurs, forming recombinant vector in the mixture. The next step is the transformation of recombinant vector into a suitable host cell. The recombinant vector is transformed into a suitable host cell such as bacterial cell. Some bacteria such as Bacillus, Haemophilus, and Helicobacter pylori are naturally transformable. They take up recombinant vector automatically.
So the third genetic engineering technique that we will be going to talk about is what we call the gene splicing. Now, before we move on to gene splicing, let us first define what is a gene. Well, genes are the DNA sequences that code for protein. So gene splicing is a form of a genetic engineering technique where specific genes or gene sequences are being inserted into the genome of a different organism. It can also be specifically referred to a step during the processing of deoxyribonucleic acid or DNA to prepare it to be translated into protein. Okay, for us to fully understand what is gene splicing, I have inserted here a summary wherein the DNA is being cut out of the organism and being put into another organism. So in here, a trait will be transferred from one organism to another. Like for example, the human insulin gene can be removed from a human cell. Then it is being inserted into a bacterial cell. Now, the bacterial cell will make or produce human insulin. And it is an example of gene splicing. Okay, next, we will be going to tackle how are genes being cut for gene splicing. And we will be needing two. These are bacterial plasmid and the restriction enzyme. Let us tackle them one by one. A bacterial plasmid is a circular DNA in a bacterial cell. and it is said to be very simple and easy to manipulate, while in the restriction enzyme, it is the enzyme that cuts the DNA at a specific code, and this enzyme can be like a photocopy machine of the gene or sequences. Of course, when we are talking about gene splicing, those two, the plasmid and the restriction enzymes, are very important. Now, how is gene splicing being done? First, of course, the restriction enzyme cuts the insulin gene out of the human DNA. Then after that, the plasmid is removed from a bacteria and being cut with a restriction enzyme. Third, the human gene is placed into the bacteria plasmid. And lastly, the plasmid is being placed back into the bacteria. Now, the bacterial cell has directions to make a human insulin. And the term now for this is transformation. It is because when a gene from one organism is being transferred to different organism, and the organisms that have DNA transferred to them is what we call transgenic organism. So trans means different, while genic refers to genes. So it is transgenic organisms. So for the next part of our report, we have provided here an examples of transgenic organisms. We have provided transgenic animals, transgenic bacteria, and transgenic plants. Let us discuss them one by one. For transgenic animals, these are genes that are being inserted into animals so they produce what humans need, like for example, the transgenic cows. These are for the cows that are being inserted gene that increases milk production. While the glow-in-the-dark cats, scientists made use of a virus to insert DNA from jellyfish. So the gene that made the cat produce a fluorescent protein to its fur. Second is the transgenic bacteria. It is a gene being inserted into bacteria, so they of course produce things that humans need, like for example, the insulin and clotting factors in blood are now made by bacteria. And the last one is the transgenic plants. These are plants that are given genes so they meet human needs again. Like for example, transgenic corn. This is given a gene so corn produces a natural pesticide. So now, they don't have to be sprayed with cancer-causing pesticides anymore because of the gene being inserted to the plant or to the corn. So now, let's move on to gene therapy. It is a technique that modifies a person's genes to treat or cure diseases. So with gene therapy, doctors deliver a healthy copy of a gene to cells inside the body. This healthy gene may replace a damaged or mutated gene, inactivate a mutated gene, or totally introduce an entirely new gene. So carriers, called vectors, transport these healthy genes into cells. But since the 1990s, gene therapy has been used in clinical trials to treat diseases and conditions such as AIDS, cystic fibrosis, cancer, and high cholesterol. But this technique is not approved for human use yet, as there are some possible side effects. And the next type of genetic engineering technique that we'll be going to tackle is gel electrophoresis. What is gel electrophoresis? Gel electrophoresis is a procedure used to separate biological molecules by size. And separation of these molecules is being achieved by placing them in a gel with small pores and creating an electric field across the gel. So the molecules will move faster or slower based on their sizes and electric charges. So the purpose of gel electrophoresis is to visualize, identify, and distinguish molecules that have been processed by a previous method such as PCR, enzymatic digestion, or an experimental condition. And the next part of our report is all about the process of gel electrophoresis in genetic engineering. What's the process? How is gel electrophoresis being done? And these are the steps. First, of course, the restriction enzymes has a role in it again, just like in the gene splicing. They are responsible for cutting or cleaving DNA into smaller segments of various sizes. Next, the DNA segments are loaded into wells in a porous gel. And a gel is spongy and the DNA squeezes through the pores. Now, the gel floats in a buffer solution within a chamber between two electrodes. And then after that, an electric current is passed through the gel. The negatively charged DNA fragments move towards the positively charged anode. So as the DNA fragments move through the gel, the smaller fragments move quicker and farther than the larger DNA fragments.
Let us now move forward to the different uses of genetic engineering in different fields. First, in the field of agriculture. Genetically modified crops produce higher crop yields because of their certain traits such as insect resistance. So this trait provides farmers with season-long protection against target pests. This reduces the need for pesticide applications and lower input costs. So how does this work? So it works by genetically inserting the Bt proteins. Bt is a protein that is toxic to specific insects. Bt genes are engineered into plants to make crops resistant to insects. These are many Bt proteins, but their mechanisms of action are similar. First, the Bt protein, shown in blue capsules, is absorbed by its insect-specific receptors in the lining of the gut. And then the second one, the activated proteins, as shown in the green capsules. So these form a complex which bores into the gut of the insect, causing the content of the gut to spill out. This eventually kills the insect. So some of the pests that are targeted by insect-resistant crops are the corn rootworm, cotton bollworm, and the soybean looper. So the modified crops are the corn, cotton, soy, and eggplant. So the trait features of these GMOs are that all Bt proteins are derived from the soil bacteria Bacillus thuringiensis. That's why it is called Bt protein. So reduces yield loss to pests, fungal toxins from infection, pesticide use, and the carbon footprint associated with pesticide application. Furthermore, it targets specific insect species due to lock and key interaction with gut receptors. And this does not have health impacts in birds, fish, or mammals. And last trait, this GMOs requires resistance management to reduce the risk that pest resistance to trait will proliferate. Next one, drought resistance. So GM crops that express drought resistance can grow in much drier areas, conserving water and other environmental resources. So the picture here illustrates the effects of drought on soybean and the breeding drought smart soybean. So in the upper part, the effects of drought on soybean, So there are changes and many effects of droughts and soybeans. Some are morphophysiological changes. This reduced leaf surface area, reduction in plant height, decreased branching, and reduced chlorophyll. Also, another effect of drought in soybeans is it causes osmotic adjustments. So it increases sugar content. It also causes the synthesis of osmocytes such as proline, betaines, sugar alcohols, and organic acids. And also, there are changes of root architecture. Because of this, it changes in root length, more roots in the surface area, and the widening of the root angle. Also, drought affects the plant because it damages its oxidation. So the accumulation of free radicals or the reactive oxygen species or ROS. So it also causes a synthesis of enzymes such as the glutathione reductase. And lastly, the drought affects the soybean because it causes yield loss. So there will be decrease in pod number, poorly developed pods, reduced seed weight, reduced seed number, and decline in seed quality. Moving on to the upper part of the picture, the breeding drought soybean. So in the right picture, we can see the drought susceptible, and on the left side we can see the drought smart soybean. So we can see a visible difference, which is the drought smart has a higher survival in drought. They are healthy, and while the drought susceptible are dying. So there are different strategies to breed smart drought soybean. These are advanced phenotyping and breeding, marker assisted breeding, genetic engineering and genome editing, and also microbial inoculants. So another use of genetic engineering in the field of agriculture is it produces herbicide-tolerant organisms. So crops that can tolerate specific herbicides allow farmers to fight weeds by applying herbicides only when needed, and enable them to use no-till production methods that preserve topsoil, prevent erosion, and reduce carbon emissions. So herbicides are the agents that are used to destroy or inhibit plant growth. So we can see here in the picture that the normal cell activity and the transgenic type plasmid. So when the transgenic type plasmid is inserted into the plant, when it's sprayed with glyphosate, the transgenic EPSPS is resistant to herbicide, so the plant will remain active. While when the plant isn't injected with this and have a normal cell activity, when sprayed with glyphosate, the endogenous EPSPS inhibited by herbicide will cause the plant to die. Next one, disease resistance. Through genetic modification, the Hawaiian papaya industry was able to recover from the devastating papaya ring spot virus that had crippled the industry. So the virus resistance worked in 3 steps. In the case of papaya, so first step is a gene from a virus such as the papaya ring spot virus is engineered in the crop and is expressed forming segments of double-stranded RNA. So the dsRNAs, or the double-stranded RNAs, trigger the plant's defense mechanism and are cleaved into short RNA molecules. Then the last step would be the short RNA molecules are complementary to the RNA from the virus. So this trigger the plant's defense. This trigger the plant's defense mechanism and prevent the virus from replicating. So if the virus will be prevented from replicating, the virus will start to degrade. So the crops that are available on the market that have virus resistance are papaya and squash. So these crops have the traits that reduces crop loss and food waste, and it also reduces pesticide use and expenses associated with controlling the spread of the virus. And crops do not require any particular farming practices. So this crop also have protection in the specific and may not protect against related viruses. So they have only protection against the specific viruses, such as the papaya, the ring spot virus only. So there are multiple ways to create the short RNA molecules, including the use of RNAs that have a stem loop structure. And the second field, the field of livestock. So genetically engineered farm animals are aimed at improving animal productivity, food quality, and disease resistance, and environmental sustainability. For example, pigs have been genetically engineered to express the delta 12 fatty acid desaturase gene from spinach for higher levels of omega-3, and goats have been genetically engineered to express human lysozyme in their milk. Such advances may add to the nutritional value of animal-based products. Also, in livestock, genetic engineering is used in research animals. So biomedical applications of genetically engineered animals are numerous, and these include understanding of gene function, modeling of human disease to either understand disease mechanisms or to aid drug development and xenotransplantation. So during trials and the development of different drugs, the research animals are the first one who will try this, if they are effective or not. So if they are not effective, it will not move into a higher phase of the trial. And then the other picture shows the xenotransplantation, or the transplantation of like this case, the donor will be from an animal, the swine, and the receiver will be a human. So since there are many diseases that needed transplantation, and the donors are limited, the researchers find a way to have transplantation through having animal donors and transfer them into humans. So swines are anatomically and physiologically analogous to humans. That's why they mostly pick this organism in transplantation. In the field of industry, so industrial applications include transforming microorganisms such as bacteria or yeast, or insect mammalian cells with a gene coding for a useful protein. Mass quantities of the protein can be produced by growing the transformed organism in bioreactors using fermentation, then purify the protein. Additionally, its application also includes producing enzymes for laundry detergent, cheeses, and etc. So as the demand of these products increase, the resources will go low. So genetically engineering these microorganisms like the bacteria or yeast will hasten the production of these products, so it will meet the demands of the community, and it is vital for our survival and for our everyday lives. In the field of medicine and health, so genetic engineering are used in the production of vaccines, in productions of hormones, and in gene therapy. So in vaccines, the use of modified yeast cells to produce vaccines against the hepatitis B virus. So there are diseases that are needed to be addressed, and thankfully we have this technology right now, so researchers and professionals can now genetically engineer vaccines to prevent and to treat diseases like hepatitis B virus. So in hormones, bacterial cells are used to produce human insulin. So diabetes is a fatal disease that when left untreated, it will just cause the human body to deteriorate and eventually die. So this will help the body to produce the hormones to be created most specifically in the pancreas. So gene therapy, modifying human DNA to repair or replace a faulty gene. So gene therapy is being used to correct the mutations, the DNA that are altered. To correct it, they are corrected, and then the process of that will be the cells will be removed from the patient and then genetically modified and engineered, usually through a vector, and then replaced and reinserted into the patient's body. For example, the field of medicine and health, one of its earliest uses in pharmaceuticals was gene splicing to manufacture large amounts of insulin, made using cells of E. coli bacteria. Interferon, which is used to eliminate certain viruses and kill cancer cells, also is a product of genetic engineering, as are tissue plasminogen activator and urokinase, which are used to dissolve blood clots. So another byproduct is a type of human growth hormone. It's used to treat dwarfism and is produced through genetically engineered bacteria and yeast. So just like in the picture below, if the growth hormone will enter the liver, it will produce IGF-1, and then when it will go to the bones, it will have an indirect effect of increase in bone length. And when it comes to the fat, if the growth hormone is inserted into the fat, it will have a direct effect of shrinkage of the fat. Upon doing the report, we have drawn these conclusions. Genetic engineering has emerged as a vital component of modern life, and its numerous implementations have grasped and morphed nearly every major domain required for survival. This innovation has revolutionized the medical, agricultural, and pharmaceutical industries. Genetic engineering is a set of techniques that are used for a variety of purposes depending on our needs. This technology has cured many diseases thanks to pest-resistant crops, transgenic organisms and hormones, and other vital products. Without this technology, the current world of biological research is fragmentary. Genetic engineering is the remedy to all of our current problems. This technique is the game changer in almost every industry, and with the passage of time, it could lead to solutions to all of society's ills. That would be the end of our report. Thank you.